In this session, we are going to discuss about life cycle of an angiospermic plant. Angiosperms are flowering plants that are classified based on characteristics that include cotyledon structure, pollen grains, as well as flower and vascular tissue arrangement. Monocots contain a single cotyledon and have veins that run parallel to the length of their leaves. Their flowers are arranged in 3 to 6-fold symmetry. Dicots have flowers arranged in whorls, two cotyledons, and a vein arrangement that forms network within their leaves. The life cycle of angiosperms, like all land plants, alternate between diploid sporophytic generation and haploid gametophytic generation. So angiosperms represent the extreme end of a trend in the evolution of vascular plants. The sporophytic generation becomes larger and more independent of the gametophyte, while the gametophyte becomes smaller and more dependent on the sporophyte. In angiosperms, the sporophytic phase is the dominant phase in the life cycle. The angiosperm life cycle occurs through various stages like microsporogenesis, microgametogenesis, megasporogenesis, megagametogenesis, double fertilization and triple fusion, then embryogenesis, endosperm development, seed formation, and fruit development. The flowering plants produce haploid spores. In microsporogenesis, the microspores are produced within the anthers. In megasporogenesis, megaspores are produced within the ovule. In the microgametogenesis, the microspores divide to produce the male gametophyte, that is the pollen grain. In megasporogenesis, a megaspore develops into a female gametophyte within the ovule, and the ovule is retained within the ovary. Within the female gametophyte, an egg cell develops. During pollination, the pollen is released and distributed by wind or other vectors or agencies like bees, butterflies, etc. Some may end up on the stigma of receptive flower. The pollen germinates to produce a tube which digests its way through the stigma and down to the style. The pollen tube grows, carrying nuclei towards the ovary and reaches the ovule. Once the pollen tube is near the egg, the two sperm cells are released. During double fertilization and triple fusion, an egg and sperm fuses to produce a zygote. The second sperm fuses with the primary endosperm nucleus within the female gametophyte, and this produces a tissue called angiosperm. During embryogenesis, the zygote develops into the embryo. The ovule becomes the seed, and outer part of the ovule, that is the integument, becomes the seed coat. The seed germinates and embryo develops into a new sporophyte. This is the diagrammatic representation of the life cycle of an angiosperm, which includes the steps like microsporogenesis, microgametogenesis, megasporogenesis, megagametogenesis, double fertilization, then triple fusion, embryogenesis, endosperm development, seed formation, and fruit development. This is the sporophyte, which produces the flowers, flowers with anthers, that is the main reproductive structure, and this is the pistil. Anthers are nothing but the microsporangia, which produces the microspore mother cells. After meiosis, it produces microgametophyte through microgametogenesis, and germination of the pollen grain with the two male nuclei forms the male gametophyte, whereas here ovary with ovules, macrospore, then meiosis, formation of megagametophyte, that is the embryo sac. So here after pollination, fertilization takes place, which leads to the formation of embryo, endosperm, and later seed setting and fruit development. After the release of the seeds, they germinate and produce a new sporophytic plant body, and this is how the life cycle completes. The homologies with respect to the terminologies: carpel, called as the megasporophyll, ovule, megasporangium, embryo sac, female gametophyte, egg, female gamete, stamen as microsporophyll, anther, microsporangium, pollen grain, microspore, and germinating pollen grain is the male gametophyte. Thank you.